find hidden patterns in genes. Imagine you've just run a genetic experiment, maybe RNA sequencing on patients with a disease, and your computer spits out a list of hundreds of genes that look important. The problem? That list is long, messy, and overwhelming. What do you actually do with it? This is where scientists play detective. Instead of staring at a jumble of gene names, they use powerful tools like pathway enrichment analysis and gene co-expression networks to uncover the hidden biological patterns in that chaos. Think of genes like people in a city. Alone, one person's actions might not tell you much, but when you see dozens of people suddenly gathering in the same place, say at a stadium or a protest, you know something significant is happening. Genes behave in the same way. If lots of them show up together in the same biological pathway or consistently move in sync across conditions, it's a sign that something deeper is at play in the disease or system you're studying. So the first thing that we are going to explore in terms of a methodology to sort of dissect our gene list is pathway enrichment analysis. So what is pathway enrichment analysis? It's a way of asking, among all the genes I found, are any biological pathways showing up more than I'd expect by random chance? A pathway here just means a set of genes that work together in a process, like the interferon signaling pathway, which controls antiviral defenses, or glycolysis, the pathway cells use to make energy. To do this detective work, you need a map of all the possible hangouts. Scientists rely on databases that list known pathways such as the Kyoto Encyclopedia of Genes and Genomes, which maps pathways like metabolic cycles and signaling cascades. The Reactome is a curated, detailed map of human biological pathways, and gene ontology are not exactly pathways, but structured categories of what genes do, where they act, and how they interact. So once you have your suspect list of genes and your pathway maps, you need the software to compare them. In terms of software analysis tools, some of the most popular ones are Decode, which is one of the earliest web tools and is still widely used today. Enrichment, a user-friendly online tool that quickly checks your gene list against hundreds of databases. And gene set enrichment analysis, often considered the gold standard. It doesn't look at what genes are significant, but at how the entire ranked list of genes trend together. And lastly, Cluster Profiler. It's an R package that lets you do enrichment inside your analysis pipeline. Each tool has its own strengths. Decode and Enrichment are simple and fast, while GCNs and Cluster Profiler are more detailed and rigorous. Let's make this more concrete with an example. Let's take the lupus pathway enrichment example. Say you're studying lupus, which is an autoimmune disease. Your RNA sequence experiment gives you 200 genes that look different between patients and healthy controls. On their own, they're just names: STAT1, IRF7, TNFA, IP3. But when you run enrichment with GCN, suddenly you see that many of them belong to the interferon signaling pathway. That's the same pathway our cells use to fight viruses, and in lupus, it's often overactive. Now the story clicks. Lupus isn't just random. It's tied to a runaway antiviral defense system. This shift is powerful. Without enrichment, you just have a long list of genes. With it, you have a biological narrative, a storyline you can test, validate, and maybe even target with therapies. But what if patterns isn't in the known pathways at all? Pathway enrichment is limited by what scientists have already discovered and annotated. What if your genes are connected in ways no one has mapped yet? This is where gene co-expression networks come in. Gene co-expression networks, instead of starting with a database, scientists look at the data itself. So with this exact methodology, genes, we look at which genes always rise and fall together across dozens or hundreds of samples. Which ones form type modules of coactivity. The logic is simple. If two genes are consistently expressed together, chances are they're working together, even if you don't know yet how. To map these hidden neighborhoods, scientists use tools like weighted gene co-expression network analysis, which is a popular R package that builds networks of co-expressed genes. And with tools like Cytoscape, this is a visualization software that lets you see networks of genes as maps with clusters, hubs, and edges. These tools turn raw gene expression data into interactive maps, where clusters of genes emerge like neighborhoods in a city. For example, in cancer research, this approach has revealed hidden gene modules that drive tumor growth. Even when no known pathway was involved, co-expression networks, or this methodology, showed groups of genes firing together, pointing to new biology, and sometimes brand new therapeutic targets. So in the end, scientists use two detective tools, and that is pathway enrichment and co-expression networks. With pathway enrichment, in summary, it allows us to see where known biology is lighting up, whereas with co-expression networks, it helps us uncover hidden neighborhoods we didn't know existed. Together, they transform overwhelming gene lists into meaningful stories about health and disease. So the next time you hear about researchers finding a new gene signature for a disease, remember, they're not just hunting for individual genes, they're uncovering the hidden pathways and the secret networks that shape the story of our biology. If you learned something new in this video, please make sure to leave a like, subscribe, and support us on Patreon so we can continue making videos like these.